college. What should a premier educational institute like IIT Madras be doing? And NPTEL is an answer to that.
NPTEL is an online courses or a MOOCs platform. We can call it a revolution in online education.
Today, nptel.ac.in, our website, is one of the largest repositories of engineering content in the world.
The type of students that we are able to reach is a fairly broad spectrum which goes into this idea of lifelong learning.
NPTEL with the local chapters is actually a very collaborative environment in which we want to cooperate and work with the colleges rather than being a competition for them.
Wherever you are, whoever you are, if you're interested, you can learn.
The Institute of Eminence scheme has been launched by the Government of India to empower higher educational institutions and help them become world-class teaching and research institutions. IIT Madras is proud to have been selected as one of the select institutes of excellence. As part of our commitment to the cause of fostering world-class research, IIT Madras has set up a number of research initiatives in diverse fields of contemporary relevance. Many of these initiatives will go on to become centers of excellence within the IIT Madras system. A total of 68 research initiatives belonging to 21 identified technology clusters are presently underway. As part of these initiatives, it is proposed to host a series of webinars from each cluster to showcase the innovative research being generated to various stakeholders like researchers, industrialists, and policymakers. These webinars will give an opportunity to engage in conversation with eminent faculty from IIT Madras and other international researchers, and also to find more collaboration in those research areas. So why wait? Click the link below to be a part of this.
Although electric vehicles or EVs are all the rage today, India still imports most of the batteries required to run any EV. This is finally changing. Indian government has recently laid down encouraging policies to move from petrol and diesel-based vehicles to electric vehicles, and research at CB will have a direct positive impact on this shift, making India and this world a greener place.
With IIT emerging as one of the top engineering institutions and educational institutions, the hospital staff take pride in telling you that we maintain the health of all the staff and students. The Institute Hospital has about 11 doctors, 14 staff nurses, 8 nursing helpers, and a host of administrative staff, and we have outsourced our departments, the pharmacy, the lab, as well as the ambulance and housekeeping services. With an aim to maintaining quality of our care, we have accredited ourselves with the ISO 9001-2015. We maintain the holistic health of all the staff, the students, and the project staff who come to us, making sure that they get good evidence-based medicine.
Thank you, Shivram, for those lovely videos. Uh, Let us start. So, a very good evening to everyone who virtually present through Zoom and YouTube. So, this is Rudra from the Office of Global Engagement, IIT Madras. As we all know, the Institutions of Eminence scheme has been launched by the Government of India to empower higher educational institutions and help them become world-class teaching and research institutions. IIT Madras is very proud to have been selected as one of the select institutes of excellence. So a total of 68 research initiatives belonging to 21 identified clusters are presently underway. As a part of these initiatives, it is proposed to host a series of webinars from each cluster to showcase the innovative research being generated to various stakeholders like researchers, industrialists, and policymakers, and even students. And that's how the Iris webinar have started and running very successfully. So it's my pleasure to welcome you all for this 34th webinar of this Iris webinar series. So today's webinar is on micro nano biofluidics under the cluster medical technology. So this research initiative project is being led by Professor Ashish Kumar Sen. So to introduce Professor Ashish Kumar Sen, Professor Ashish is a professor in the Department of Mechanical Engineering at IIT Madras. His research interests are in the areas of micro, nanoscale, interfacial phenomena, and then lab on chip technology. He has received several awards and fellowships such as Swarna Jayanti Fellowship, Institute Research and Development Awards, mid and yearly career levels, NASI, Scopus Young Scientist Award, Young Faculty Recognition Award, and Indian National Academy of Engineering and Young Engineer Award. I mean, I have a lot more to name. Joining as the speaker of today's program, we also have Professor Thomas Laurel, who has joined as the moderator of today's session. Professor Thomas is a professor in the medical and chemical microsensor at Lund University. He has received several prestigious awards such as the Axo Nobel Science Award for his groundbreaking disciplinary research, Wallmark Award by the Royal Swedish Academy of Sciences for his microsensor system innovations, Scapa Award in the memory of Alfred Nobel for the best innovation, and also Erna Ebling's prize for the outstanding research in the microfluidics and biomedical engineering. He is the founder of 4 startup companies. He is one of the founders and the past presidents of the Chemical and Biological Microsystem Society. It's my pleasure to welcome you, professors, to this session. A note to participants, please use the Q&A to enter your questions and upvote the question that interests you so that the moderator can prioritize them. Over to you, Professor Thomas.
So I understand that maybe I should just briefly say a few words, and I would be very happy to invite Professor Ashish Kumar Sen to come and present the new center that is a fantastic achievement that he now will present to us. And I will also be happy to discuss the outlook for the center and the research field at large for the audience here, and I'd be happy to take questions, and if they come in the Q&A, I will be keeping an eye on that after Professor Sen has given his presentation. So please, Professor Sen, go ahead.
Thank you, Professor Laurel, for your time in moderating the session, and thank you, Rudra, for the kind introduction. And let me welcome the audience to this talk. So our project is titled Micro Nano Biofluidics. Here is an overview of my today's presentation. I would quickly introduce the team and collaborators, and then briefly talk about the background of Micro Nano Biofluidics, and then present briefly about some of our research activities. So we are a team of 10 faculty members from different departments at IIT Madras. We also have close to 25 PhD scholars and 6 project staff as part of the team. We have highly accomplished researchers from all over the world as academic collaborators. And we also have 6 industrial collaborators as part of this effort. Now, to talk briefly about the background, if one talks about biofluids, in particular blood, which is a very common biofluid, one sees particles of different sizes. So you have cells which range anywhere from 1 micron to about 20 micron. And then if you look into some of the cells, you find DNA, and the suspending fluid for these cells also have contain plasma proteins. And the DNA and proteins are typical sizes of tens of nanometer to hundreds of nanometers. Apart from blood, you have other bioparticles which are of interest as well. You have bacteria which have size range anywhere from 1 micron to 10 micron. And then you have virus, which is something being talked about a lot in the current scenario. So which has size anywhere from 10 nanometer to about hundreds of nanometer. Then you have sperm cells, which have a head, which is about 5 cross 3 micron, and a long tail, which is about 50 micron long. And then you can also talk about C. elegans, which are excellent model systems because they have a genetic characteristic similarity with that of human being. So these C. elegans of size width of about 100 micron, and they're close to a millimeter long. So what we see that these bioparticles are mostly a micro nanoscale dimension, and this is one of the motivation why we are trying to understand micro nanosystems. And these biofluids which suspend these particles are viscoelastic, and which makes the study even more challenging. One could attempt to understand the behavior of these biofluids and particles through understanding of micro nanoscale physics. If one knows the length scales are for these bioparticles, one could also look at the time scales. And as you can see, the time scales and length scales are intimately related. So what you see here, things that are smaller, process for them happens to be faster. For example, if you are looking at micro nanofluidics, you talk about length scale somewhere in the range of a millimeter to about nanometer. And the corresponding time scales will be of the order of millisecond to about nanosecond. And such systems are typically studied in microchannels, which have dimensions of the order of 100 micron or so, and or in nanochannels, which have a dimension of the order of 10 nanometer. And what you see here is generic architecture of a micro nanofluidic device. So what we have is fluidic space, and which is miniaturized. Then you have exposure to external field, be it electric field, magnetic field, or acoustic field, and so on. And then you also have a mechanism to detect, interrogate, measure whatever function is happening inside the device. There are several advantages of these micro nanofluidic systems. First being the low sample consumption because of the miniaturized nature of the space, and which leads to also smaller reagent consumption. Because these devices are automated, one could also parallelize processes and look at high throughput experimentation. And one of the unique features of these devices is the high surface area to volume ratio, which brings in very unique physical phenomena that are normally not observable in at larger length scales. By understanding all these aspects, one could develop lab-on-a-chip systems. For example, if you talk about a typical pathological lab, you lyse samples, you can dilute sample, you can purify, you can elute and detect. The idea is all these functionalities can be really squeezed into a typical credit card size chip, and which will have different modules to carry out all the function that you do in a typical patholab in wet benches using large instrument on a single chip. And that you do at a much faster speed and requiring a smaller amount of samples and reagent. And you also have a system which is automated. So that is what is brought by lab on a chip. Now, if you talk about the focus of our group, the focus is really into two different aspects. One is micro nanoscale interfacial phenomenon, where we focus on the physics side of micro nano biofluidics, and then develop a lab on a chip technology for specific applications. In the micro nanoscale interfacial phenomena, our focus is really to understand behavior of particles, cells, fluid interfaces, and droplets. And one of the techniques that we use to achieve this is through application of sound waves, which we call as acoustic fluidics. Typically, if you have a channel and you have particles in the channel and you expose this channel to standing wave, these particles would experience what is known as acoustic radiation force, which is given by this expression. A is the particle size, E is how much acoustic energy you actually put it in the system, K is the wave number, and phi is a parameter that actually is a function of the property of the particle and the medium. If the acoustic impedance, which is density times speed of sound of the particle, is greater than that of the medium, these particles end up at the nodal plane, and if the particle has a lower impedance as compared to the medium, they end up at the antinode. Now, apart from these, the primary force that this particle experience, because a particle can have other particles in the surrounding which can scatter the sound wave, it can lead to secondary radiation force. One of the problems that is of our interest is to look at behavior of dense suspension. So these are typically suspensions which have volume fraction greater than 0.1. And our studies have shown that the behavior of this dense suspension is actually markedly different compared to dilute suspension, where the volume fraction would be much smaller than 0.1. And our study has shown that if you are looking at focusing of such dense suspension, the interplay between acoustic radiation force and the shear-induced diffusion force, which is a function of how dense the sample is and how fast the sample is flowing in the channel, becomes very critical. And since you are talking about dense suspension, where one particle can have surrounding particles, the interparticle force comes into picture, which we have also studied in one of the recent work. And one can in fact talk about a dense suspension having particles of two different sizes or many different sizes. And as you have seen, these bioparticles are non-spherical objects. And some of these particles are even motile. They can self-propel themselves. And they are suspended in viscoelastic fluid. And all these aspects make the problem very rich and very challenging. And one needs to understand all these aspects if one wants to completely understand such a problem. Another interest we have is to look at laminar co-flow system. And when we expose this to sound wave, and what we have observed is that if you have such a co-flow system and you expose this to sound wave, like particles, the high impedance stream actually tends to relocate towards the nodal plane. And this happens when the acoustic energy is sufficient, so that the acoustic force overcomes the interfacial tension force, because you have a clear interface between these co-flowing streams. And this stream to central thread transition was observed when the capillary numbers of the stream is greater than 1. And it was found that this process is in fact reversible. That means when you turn on the sound wave, you see the stream going as central thread. And when you switch off the field, it actually goes back to the original co-flow system. When the capillary numbers are smaller, you observe even something more interesting. When these high impedance stream tend to relocate to the center, it actually breaks into small droplets. So one can achieve from stream regime to droplet regime. And by switching on the field, one can in fact see that the original co-flow regime will re-emerge. So one can undergo transition from stream regime to droplet regime by switching on and off the acoustic field. On a more fundamental level, one needs to understand how these acoustic standing waves are formed, because you have two immiscible stream, and for a generic co-flow system, formation of this standing wave is yet to be understood. And one can also understand, since you are able to generate on-demand droplets, how we can selectively encapsulate cells and sort them. In addition to our studies of these interfaces inside channels, we are also interested to look at interfaces on surfaces. For example, here, our objective was to how we let a drop spread on a superhydrophobic surface, which is typically difficult to achieve by applying sound wave. And we have also studied how a pair of liquid drops would interact on a liquid infused surface, how they would repel each other and how they would attract each other, eventually leading to coalescence of drops. And one can also study motility of drops on tracks that have wettability gradient. So these drops can self-propel themselves. And when they arrive, if there is a junction, they can also split into multiple droplets. And one is in particular interested in such phenomena because they can lead to a technique where one can carry out biochemical assays on surfaces. Besides this, we are interested in isolating circulating tumor cells from blood cells, both using labeling techniques as well as label-free techniques. For example, here in this work, our effort was how to isolate these target cells, which are circulating tumor cells, from the background peripheral blood mononuclear cells in single cell format. So what we do is we encapsulate these cells inside droplets, and these cells are tagged with fluorophores which are specific to these cancer cells, typically the EpCAM markers. And once we read a target cell, it actually activates a coalescence module downstream. We selectively isolates the background cell from the target cell, which is then captured inside droplets for downstream single cell analysis. We have also looked at level-free techniques for isolation of CTCs. For example, in this work, our objective was to isolate circulating tumor cells from peripheral blood mononuclear cells by exploiting the difference in acoustic impedance. In a situation where the acoustic impedance of CTCs is higher compared to that of PBMCs, these are infused into the side stream, and we take care of the impedance of this medium such that the acoustic impedance of the medium and the PBMCs match. So the motion of the PBMCs is arrested, and only the circulating tumor cells which have higher acoustic impedance, they migrate towards the nodal plane and they get separated. And you can do exactly the reverse if you are expecting that your CTCs have lower acoustic impedance compared to PBMCs. So this is an ongoing effort of achieving better and better techniques for CTC isolation. So for that, it becomes important that you understand cells on a more fundamental level, which we have been trying to achieve through experiments as well as simulations. So for example, we have been trying to use optical twitching technique to probe into individual cells to observe how they move. For example, the pitch motion of cells is what we are interested in. And we are also interested in understanding cell behavior, how they interact with the surrounding medium, how they behave under confinement using molecular dynamics simulation. The other interest that we have is to develop lab-on-a-chip technology for measuring level of gasotransmitters. As you know, gasotransmitters participate very intimately with the physiological states of body. And the idea was by measuring the level of gasotransmitters, whether we'll be able to predict onset of systemic inflammation. So this system has various models. The idea is to extract the plasma in which we are going to look at these gasotransmitters and then have them interface with specific chemical probes that we are designing in collaboration with chemistry to interface with each one of these gasotransmitters, which then generate fluorophores, and that we read using laser-induced fluorescence technique. We also have interest in developing lab-on-a-chip for detection of dengue analyte. Here we are talking about a device which is standalone. It doesn't require external energy for sample infusion and plasma separation. And once this plasma, which contains the analytes, are separated, we then look at localized surface plasma resonance technique, which uses a substrate having nanostructures which are functionalized against antibody, which are specific to these analytes that we want to detect. And when the plasma actually runs over this functional light surface, the refractive index around these nanoparticles change, and which gives rise to a shift in the peak absorbance wavelength. And the shift in the wavelength is actually related to the concentration of the analyte in blood. And right now, our effort is to develop an integrated device to be able to detect NH1 antigen, IgG, and IgM, to be able to detect dengue from blood sample. With that, I would stop. There would be opportunity if you are a student and if you are interested to join our group. I would encourage you to visit our website, cmnbf.in, and you are also welcome to write emails about your interest. So with that, I would stop and thank you all for your attention. I'll be happy to respond to your questions.
Okay, thank you very much. Impressive overview of the activities that you have set up. And when I visited you a couple of years ago, I can see very distinct developments ever since. And so I'm quite impressed of the bandwidth of the research that you're doing. And I would just like to take the opportunity to discuss with you a little bit few more general topics, and maybe at the end also jump in on some more detailed questions. In the meantime, maybe our audience can think of questions they'd like to give you specifically, and we could jointly see if we can answer them. So I can just reflect a little bit of the field that you are working in, and more generally in the life science field, that we over the past year have seen a fantastic development within the biosciences, and partly driven then by the lab on the chip field, not only, of course, but as one component, important component. So for instance, in the early 2000s, the full sequence of the human genome was presented, both in Science and Nature, as a major global effort over 15 years to try to sequence the human genome. And now, 20 years later, we have the capacity to sequence the human genome 30 times over in one single instrument in a laboratory. So this gives a perspective over the tremendous development we have seen, which is a combination of smart biochemistry, microfluidics, physics, optics, computer science, and data management. But altogether, I think it's a fantastic time era that we live in, and the opportunities going forward are just enormous. But
I agree with you on that, Professor Laurel.
Yeah, so that was just sort of an introduction to my sort of extreme interest of the field. And though microfluidics still a rather young science discipline, and now you have established a micro and nano biofluidics center, I think it's very timely. But can you elaborate a little bit maybe on your view of the future development of the microfluidics and nanofluidics field? Which direction will it take? What areas do you think will have most impact, and so forth?
Yeah, thank you for that question. I completely agree with you how the developments happened with respect to the genome sequencing. I would say that microfluidics had a critical role to play in the overall process. In fact, if one closely looks at the process of genome sequencing, starting from sample preparation to how the gene sequencing is read out, microfluidic can actually contribute at each and every step. And this has been really the case because the extraordinary speed and the benefit in terms of sample volume and automation that microfluidics brings in. In particular, I see one area where microfluidics is going to drive this forward is the ability to carry out single cell analysis. So microfluidics has come up with this special field called droplet microfluidics, which allows us to trap single cells. And as they say, every cell has a story to tell. So I think The fact that we are able to encapsulate and manipulate cells within droplets will open up opportunity where we could lyse and look at entire genome for individual at the individual cell level. And this could probably lead to improved and personalized therapy. And the other front where microfluidics comes in is the lab-on-a-chip diagnostics. And we have seen that even starting from the iStart system to of late, even if when you're talking about rapid antigen test kits for COVID, you are actually using the microfluidics technology. So this lateral flow assay is really popular with the advent of microfluidic technology. Then paper microfluidics has come in, which makes the process really cost effective. And microfluidics has in fact gone into almost all fields of sensors and actuators. So talking about environmental sensing to water quality monitoring and so on. In fact, one area that I could think where microfluidics has already started evolving is in the area of organ-on-chip. How one can self-assemble cell by cell at a single cell level and with an effort to simulate organ tissues, and which could also drive future drug developments. So I would say these are some of the areas where microfluidics has a lot to offer.
Yeah, I do agree with you to a large extent. This is really some areas where we see a booming form of activity, single cell analysis, for sure. We are already doing single cell gene sequencing and transcriptome sequencing and at large and high speed. What people now are struggling with, and where I think we can make a difference, is in actually doing protein and proteome a mapping of single cells. So this is something that I'm sure that the near future will actually bring into the labs. Tissue engineering, as you mentioned, is also a very, very hot topic. So we will see more of that come up. And of course, environmental applications, distant monitoring of the environment, etc., will also be quite critical. So I think there is a lot of opportunities out there, and more to come, I'm sure. So when running a center and starting up a center like this, and in order to make it thrive and for the research field to develop, recruitment of good students is essential. Without good students, you will not live for long. So recruitment and attractiveness. So what would you argue be to be good reasons for students to select to move in this direction in the choice of education and future expertise?
Yeah, I would say we are already starting to get very good students coming to our field, coming to IIT Madras and becoming interested to join emerging areas like micro nano fluidics. We are already seeing that happening. And as we briefly mentioned, this area has a lot to offer, and this is emerging, still growing. So there's a lot of opportunity for students to come and join this field and contribute. So this is the process that's already happening, and we anticipate that this is again going to step well even better as we go along.
Yeah, and I would also sort of like to complement that with a comment that if you look at the field, state of the art now and where we are, I would say that in comparison to electronic industry, we are now at the stage of mid-1960s, so the early stages of integrated circuit technology. And we will see something similar in development from now on in the future. So in terms of being able to find a job, in terms of being part of an extreme development line, this area would definitely be something, if I was young, I would again jump onto the train and go into this field. So when I started this once upon a time or got into it, it was still not even coined lab on a chip. So that has developed a lot since. So I would definitely encourage people, young students who would like in life science research, etc., to go into this. So if you look into the microfluidics and nanofluidics field, this may be more a sort of your view of the whole thing. Where do you see that we are less exploring the areas? So what are the unexplored areas where things may change in a sense? Where haven't we been?
Yeah, I think, with regards to microfluidics, I think the real missing link was actually the sample preparation. And a lot of effort has been put in that direction over the last couple of decades or so. And, I would still say that integration has been a challenge. I think the detection platforms are quite well developed. And with these sample preparation modules, where a lot of research has gone in last 2 decades or so, I think the focus can really be on the sample, the system integration and actually delivering something for specific applications. And again, as I said, there are a few applications which are emerging. For example, drug discovery. Drug discovery has a very long cycle time, as we all know. I think if microfluidics can offer something in that direction, either in terms of developing artificial organs or providing systems that can test efficacy of drugs with a small cycle time, I think all these could immensely benefit society.
Yeah, absolutely. The last point you made here, accelerating drug testing. And I think the field of organs on chip or tissue engineering is one of the really, really hot topics in the field right now, where pharma industry is looking very much into opportunities to advance their own position here. So absolutely. And system integration is definitely a topic. Many types of unit operations for handling fluids, doing sample preparation, doing sensing and measuring has been done, but building them together in one unit is still a challenge. And one key there, I would say, is standardization. We see very little standardization yet in the field, and that's still because it's young in terms of the industrial engagement. And I would predict that we will see much more standardization as we move into a further advanced field here. So on a personal level for you or senior scientist in the center, when embarking on a lab on a chip and microfluidics research, what strategy in building your group would you recommend? Where do you find the faculty and the students that you'd like to bring in?
So I think we are, as I said, we are looking into 2 different aspects. One is trying to understand the physics side of it, where we look at problems that are, quite unexplored. And then our effort is to, through this new understanding, to develop, new technologies that we want to develop. And so I think we are a quite young group, as you pointed out, but I think there is a lot of drive to actually, do something and deliver something. And as I said, the students that we get in are also equally passionate.
Yeah.
So I think, together we will have a very nice ecosystem which can, take up new challenges and deliver. Yeah.
And I guess that having students coming from different faculties and areas is also part of the strategy that is pursued. And I would recommend that surely see to that you have people coming in from different research areas in the group. Let's see, we have, okay, now I see the question. I didn't see the questions before here, so there's a lot of questions. So a question here, if someone has some degenerative disease related to blood or other fluid, what will be their approach of nanofluidics on them? So that's a tough one.
Well, there are more than one approach to handle this. Let's say, you have some nanoparticles which has functionalized with some antibody which can bind to specific protein markers that you are expecting blood to contain. Let's say the blood is having some bacteria and you want to flow some nanoparticle and capture these bacteria along with it. So that's one solution. And the other solution to actually, lyse the blood cell, extract the DNA, and then look at the genome. So there are many approaches to the same problem, and it actually depends on what tools and the technique that you have access to deal with such kind of detection that you want to pursue.
Okay, so here's another question from a student. I did my bachelor's in biomedical engineering from India. I want to pursue my master's in lab on a chip technology. Where can I find some college which is providing this course? Is India having something like this course? How is the industry orienting with this technology?
Well, IIT Madras has a biomedical engineering program. So welcome to IIT Madras if you want to pursue that. And we also have an ID program at IIT Madras where, if you have a nice concept that you want to pursue, and even if you don't have a concept and you have an idea that you want to work on an interdisciplinary area, you can come and attend interviews. And if you are selected, you'll have the opportunity to work with, in fact, 2 advisors from 2 different departments. So I'm sure there are many other institutes and universities in India offering biomedical engineering program, and IIT Madras certainly does that.
Yeah, so I think students will find that under the biomedical engineering program as a subtopic. So with the lab on a chip and microfluidics for sure. So here I'm jumping around a little bit. I'm a data engineer with knowledge of AI and data science concepts. I have interest in life science research projects. How can I support into these kind of projects?
Well, I think, AI data science has really, becoming a hot field in the context of biomedical engineering, micro nanofluidics. I think we see new papers coming every day. I think if you are already in this field, I think you have a lot of scope if you want to get into micro nanofluidics or biomedical engineering in general.
Yeah, and I agree. I mean, I see where I work at the Biomedical Center in Lund, at Lund University in Sweden. I'm surrounded by people who are generating terabytes of data every day. And they have, their biggest problem is not data generation anymore. It's actually processing data and find the meaning and the keys of to answer their questions in the wealth of data that they have. And AI is becoming one of the leading tools here. Deep learning and AI, everybody is asking me about that since I'm from an engineering side. I'm not an AI person myself, but I try to just bridge them over into people who can do that. So if you know AI, have an interest in it, there is a ton of applications in the life science research area. You just go up to the labs and talk to them and they will have a job for you. I have another question here. It's actually combined 2 questions. Is it possible to create chip who would detect different bacteria by a single chip? Or can we create a chip for broad spectrum detection of parasite or bacteria?
Well, you have to have, again, it depends. There are multiple approaches to detect bacteria, and it would require you to look at some kind of markers which would bind to the specific bacteria that you are looking at, right? And as I said, if you just want to have for example, a nanoparticle which is functionalized with some kind of marker which would bind to specific bacteria, and you are expecting, let's say, 2-3 different types of bacteria, and you have to have these nanoparticles functionalized with 2-3 different antibodies which would then bind, and you can extract them and analyze. or you can lyse them and look at the specific genetic markers in them, which you already know you're expecting these markers to appear. And through PCR, one can always test for those. So yeah, it is possible.
Yeah, I agree with you. I think it's possible, and the reason for them not being out there yet is maybe that in order to see them on the market, there also has to be a market, and the market has to be defined. And these developments are expensive. If you're taking one of these ideas to a commercial product, it must be sure that you can make money on it. Another question here. Painless needles have potential to replace traditional syringes. Why have they yet not been commercialized?
Well, I mean, commercialization is a very difficult task, and which we academicians do not understand very well. But what I know is painless needles have been there for quite some time. I would recall starting 2000, as early as 2000, I was seeing papers coming on painless needles. I would recollect one from MIT, which uses, dry reactive ion etching to create these straight needles. And they, in fact, did some animal trial also with those painless needles. So I think the technology has been there for more than 2 decades, but the commercial viability is another thing.
Yeah, exactly. That's an example of where the commercial constraints holds it back from actually penetrating the market. The little pain you get from a tiny needle doesn't outweigh the cost of actually you would have to pay for each one of those painless needles, nano or micro needles. That's my opinion. So I agree with you fully. So, and this is a follow-up question on the previous one, and maybe the same answer. What career opportunities are open for people who are currently pursuing life science and data science simultaneously? I think there's just enormous.
We have responded to that question. I think it's, as you rightly said, there is huge opportunity. If you have interest in the area, and if you want to, come and ask for a job, you will get it. Yeah.
And this is a very specific one, but maybe it could inspire somebody. I have done an internship on copper-silver nanoparticle synthesis, which act as an antibacterial property against multi-drug resistance, ETEC, in vitro and in vivo in mice. I want to work further with nanoparticles. Could you suggest me best part to start?
Oh, okay. Best part to start. I think you have to, the best part is to find an advisor if you want to pursue PhD. So I think you have to find a good university and an advisor that who would support the problem that you are wanting to work.
Yeah.
It's an exciting area. Yes.
Yeah. So I'll ask you a little bit about the acoustics that you were touching on in the work. Acoustic Fluidics is a rather new discipline within the micro nanofluidics field. And so where do you see the most exciting development areas within that field? I mean, acoustics allow you to manipulate matter in microscale in a very precise and elaborate way.
Yes, I think this is a very interesting field, and we are, I would say, relatively young into the field, but the field itself is not very old. So, and that is why we have a lot of exciting opportunity within acoustic fluidics. And I would say that it is probably one of the newest technique. And if you compare with, let's say, electrofluidics, magnetofluidics, acoustic fluidics, is relatively young, but within a short time, it has really, created wonderful technology. And there are some of the fascinating aspect of this technique is that it is versatile, and We don't have to have particles which are charge neutral that, for example, electrofluidics require, or requires conductive particles, or we don't require magnetic particles that magnetofluidics requires. So all we need is particles that have different properties as compared to the medium to be able to manipulate them. And so I see there are many exciting opportunities. I think a lot of work has been done with respect to cell handling, cell sorting, and handling of blood cells, focusing of cells, separation of plasma from cells. And I would see that if one wants to improve these processes, I think the physics is still rich. For example, if you look at a cell, blood cell, it is not a sphere. Whereas the theory of acoustofluidics assumes that it is, and it has a force expression which is developed for a spherical particle. So I think there is a lot of scope in terms of, deciphering the correct physics in the realm of acoustofluidics. And if we do that, I think that would probably help in developing improved design and better understanding of the system that we want to develop. And I think, again, there are areas, for example, self-assembly, and where we want to build cell by cell, some artificial organs, where acoustofluidics can also be used. So I have seen some work coming from Max Planck, where, for example, they are using optical means of self-assembly. So one can also look at acoustic means of doing this self-assembly, cell by cell, and build artificial organs. So I would say there's a significant opportunity waiting within the acoustofluidics field, and that can influence to a great extent the overall field of micro nanofluidics.
So I don't know how much time we have still to go.
I think if you are fine, you may log off, and I would briefly go over the Q&A and try to answer as many I can. The remaining.
Yeah, because there are many questions still in the chat here, but I recall we had about 45 minutes for the total session.
I think we are about time. We could probably close, and then probably I will spend about a few more minutes to go over the Q&A, and then I can sign off after that.
Yeah, so there's a lot more for you in the Q&A, and I just didn't have time to go over them all. So
thank you very much. I think we're already over time for you. So thank you very much, Professor Laurel, for your time for us.
Okay, thank you. It was extremely fun, and I wish I could meet all the students who have been active in the Q&A here. It would be great. And I wish you all luck with the Center Formation, and I'm sure it's going to be a success, because it's a field that will just explode over the next 10 years. So tell the students to go there.
Thank you very much.
Yeah, bye-bye.
Okay. Okay, so I'm addressing this question about CTCs. So yes, CTCs, as the question rightly poses, CTCs are rare. You will find a handful of CTCs in, for example, 1 ml of blood. So considering the throughput that microfluidics handles, in fact, some of the commercial systems tend to run it for about an hour to be able to run a sizable volume of blood sample to capture CTCs within statistical significance. So that's a very valid question about CTC isolation. What wavelength of light is most applicable for optical detection of components of blood, especially non-cellular components? So I think we are using about 485 nanometer excitation and 532 detection, but that will again depend on the probe that you are using and not really what component of blood that you're trying to detect. Because this optical detection is mostly going to rely on, let's say, the fluorescence method. So it would actually depend on the dye or the tagging that you are going to do. There is no simple answer to that. It's going to depend on the dye. Okay, trying to find some more question, maybe one or two question. What is the silver nanoparticle used for dengue? Yeah, this is silver nanostructures which are grown onto the silicon surface using thermal annealing, and these have sizes anywhere between 40 to 50 nanometer. What is the red line representing the detection of dengue analytes? So let me look at what the red line represents. So the red line is actually the antibody, which is bound to the nanostructure and which will then bind to the NS1 antigen we are interested to detect. So I think I have addressed most of the questions, if not all. Can we end now?
Yes, Professor.
Stop here. So let me thank the audience for being patient and asking many interesting questions. It was thoroughly enjoyable for myself. Thank you. Thank you very much.
Thank you so much, Professor. I mean, indeed to say, it was really a good session that even I've enjoyed a lot hearing you and Professor Thomas. May I request the participants to stay connected with the IRIS team so that you cannot miss the future webinars. Thanks for your participation. Have a good evening.
Thank you, Mr. Rudra. Thank you.
Thank you, Professor Ashish.
Thank you. Bye-bye.
Ms. Ujjwala, you can proceed for closure. Thank you.